Invite Dr. Anderson to come down for Evolution's Flawed principle.
So the brave ones are back. Is that it? Natural selection in action. Okay. Trying to build upon a little bit of what I started yesterday. Actually build upon a little bit of what you heard Jay talking about this morning. In fact, I'm going to go right to a quote from Jay that he said this morning. Cutting edge science, you never have to be afraid of it. Many times as creationists, we are accused of not wanting to do science, being a showstopper, if you will, in science, because it's just what the Bible says, and so science can go do its own thing. And nothing could be further from the truth, because certainly as a scientist, if I am committed to the surety that the Bible is the Word of God and therefore speaks His truth, it's going to be revealed in His creation, as He Himself even said. You know, Psalms 19 we often quote, the heavens declare the glory of God. But many times we forget to read further where it says there is no place, there is no language where God's glory is not proclaimed. And I would say that includes, almost even specifically includes, the scientific laboratory. We don't want science to stop. We want it to go on. In fact, just teasing Jerry a little bit, we love the genomic research. Man, every day gets better for us and worse for the evolutionists. So we're saying keep going, keep going. So no, we're not afraid of science. And don't let anybody tell you are. If they say that, you know that they are ignorant of what they're saying. They don't understand creation science. All right. Early in the molecular age, way, way, way back when I was a graduate student, and I used to ride my dinosaur, Michael Denton, in his book, Evolution, Theory, and Crisis, declared that life was beyond the reach of chance. Interesting situation. See, it's only moved further away. Darwinian thinking was based upon scientific ignorance. It became popularized in a time in the scientific community when they simply didn't know very much. Cells were considered bags of enzymes. Enzymes were considered simple little types of proteins. Organisms such as the human body were considered not necessarily that sophisticated. And in fact, even recently, Darwinists have still tried to make claims about all these things that are very inept and very wrong about the human body. But the truth of the matter is that every time we study and learn and understand, we gain an appreciation of how just opposite that is. And I constantly challenge evolutionists. I am not a creationist because of what I do not know. I am a creationist because of what I do know. If there was ever a time that Darwinism could explain what we see, this isn't it. Okay? That time is long gone. Genetics, biochemistry, physiology, epigenetics, all these fields of study have moved Darwinism beyond the horizon of being able to explain anything of what we see biologically. So it's only moved further away from chance. It hasn't moved closer. All right. Speaking of chance, Richard Dawkins, in his book, Climbing Mad Improbable, probably the only book he wrote that doesn't wind down to irrational screaming. Okay? It's actually not a bad book. If you're going to read any of his books, read this one. Throw the rest away, but read this one. It's not bad. He says, quote, "It is grindingly, creakingly, crashingly obvious that if Darwinism was really a theory of chance, it couldn't work." And we would agree. Okay? So we're all in agreement on that. If it was really a theory of chance, it couldn't work. Okay? Now, he's going to make argument that it's not a theory of chance. Okay? So follow this for a second. He uses as an analogy Mount Improbable. Mount Improbable represents this huge increase in biological sophistication. So it'd be like climbing Mount Everest. You start at sea level and go all the way up. And he argues, of course, you don't take Mount Everest in a single leap. Right? If you could do Mount Everest single leap, you could probably go to the Olympics and win everything they have. So you don't do that. And that's not he says that's not what evolution does either. It can't do it in a single leap. What he argues is that it's not a theory of random chance. It's a theory of random mutation plus, it is a theory of random mutation plus non-random, cumulative natural selection. That's what he says is the key for claiming Mount Improbable. It is random mutation plus non-random, cumulative natural selection. Right? He goes on to say that Darwinism then solved the problem of climbing Mount Improbable by breaking up the improbability into small, manageable parts, smearing out the luck needed, going around the back of Mount Improbable and crawling up the gentle slopes inch by million-year inch. So by his analogy, this is how you climb Mount Everest. Wouldn't that be nice? Nice little Sunday stroll, just winding around on this little country path bit by bit, and pretty soon, voila, you're at the top of Mount Everest. Didn't even know you were climbing a mountain. That's what he proposes. Interesting, he also proceeds to say only God would essay the mad task of leaping the precipice in a single bound. So he admits that. Okay? Then he goes further and says any designer capable of constructing the dazzling array of living things would have to be intelligent and complicated beyond all imagining. And again, I say, I agree. I agree. Yes. All right. Now, here's one of the interesting points. Okay? Here's what he's doing. He's saying random mutation plus natural selection gives you this God that's complicated beyond imagination. So we either have a God that's so absolutely impressive we can't even imagine it, or we have random mutation plus natural selection. Do you see a little disconnect here? A little bit of, okay, it's interesting how God has to be beyond imagining and complexity, but we can replace that with something that's very easy to imagine, very easy to understand, at least according to him. So I find a little disconnect, but he clearly is equating God with random mutation and natural selection. Now, what's he mean by natural selection? Well, the classic view to be told is natural selection is that the most fit survive, and those that are survive are the most fit. And we'll let Bill kind of give our expression for that. Right? Now, let's let Dr. Dawkins explain a little bit further what he means by natural selection. He suggests that it is a process that will sort through all the traits in a population and favor those traits that are best suited for survival. Kind of the same thing we just said, but it's maybe a little more sophisticated in sounding. All right, so what he's suggesting is that through the process of, quote, natural selection, organisms can then be those that run faster will be selected for versus those that won't, kind of like the situation of the two guys running from the bear. And this, one friend says, I don't think we can outrun this bear. And the other friend says, what do you mean outrun the bear? All I can do is outrun you. Okay, so run a little faster, gives you a little better selective advantage. Okay? Swim a little farther, fly a little higher, hit a little harder, dig a little deeper. If these traits give you a slight advantage over your neighbors, then by his understanding of natural selection, it gives you an added benefit, making it more likely that you'll survive. And that's how he's viewing, how he's posturing how natural selection works. Now let's stop and think just a little bit using Dawkins' own understanding of natural selection. Okay. He will readily admit it has no plan. Natural selection cannot, and Jay touched on this a little bit this morning, it cannot look forward and say, I want to achieve this. It cannot do that. So let's use natural selection to figure out how it would build a 747 from a car. Right? Let's say that the tires start becoming more like on a 747. Okay? Does the car now drive better? No. So what would happen? You'll take that car back to the dealer and say, this car doesn't work very good, I need a different car. So what we have is, as we start developing the traits that make the car a little bit more like a 747, it actually becomes less useful as a car, but not useful as a 747. Okay, what happens if it starts growing a little bit of wings? Now you're having a hard time keeping it on the road. Will you like that? No. So you will, as the natural selection agent, take the car back, demand your money back. This doesn't work. So what will happen is, by Dawkins' own definition of natural selection, it will work to keep the car as a car. because it cannot look forward to, if I make these changes, eventually will become a 747, which will be superior to a car. Each change has to benefit the organism in that moment. And yet what we see time and time again in the biological world, just as we see within this illustration, is that the particular change is necessary to move incremental up that man and probable step by step, do not in themselves benefit the organism until you actually get to the completion of all the steps. Very hard to get the steps in between to each be advantageous. Just like it's very hard to get each transitional stage of going from a car to a 747 to be advantageous. There would be nothing advantageous about any of those changes until you became the 747. But in the meantime, even Dawkins' own understanding of natural selection would prevent that from happening, because you make it lousy as a car, therefore it would actually die off, not keep evolving into a 747. Okay, so see, natural selection doesn't select for anything. Okay? It selects against. Even by Dawkins' own definition of natural selection, it's a calling event. It removes from. It doesn't add to. See, a few people got it. Right? The trait that natural selection is going to be working on must already be present in the population. Natural selection does not create the trait. And this is really, I think, where we run into a lot of problem with the evolutionist views of natural selection. They try, and we saw by how Dawkins was even defining it, natural selection replaces God. But natural selection has no capability to do any such thing. Natural selection is the event of removing what does not give the organism the benefit that it needs. Okay? It only eliminates, it does not create. Let's take the peppered moths. The famous peppered moths you'll still find in textbooks. We had within the moth population in London at the time these studies were being talked about anyway. You had two different types of colored moths. You had the black moths and the white moths. And by the evolutionary scenario, okay, when the buildings were of a lighter color and the trees were of a lighter color and such, the white moth had the advantage because it blended in more. The black moth stood out. Therefore, the predators, such as birds, can more easily see the black moth and pick them off and eat them. Then, supposedly by the scenario, when the soot and smoke built up in London and started covering the sides of trees and the sides of buildings and such with darker color, then we had a switch. Now the black moth begins to blend in better and the white moth stands out. Therefore, again, by the scenario, the white moth now is more subject to predation, and we saw an increase in the population of black moth. Now, there are several things to bear in mind. First, there was always a black and a white moth population. Years ago, I had a student technician that was taking evolution, and I would just ask her questions from time to time. And she was talking some about they had just studied the peppered moths, and I said, oh, okay. Now, you understand that both the black and the white moths were always there all the time. It's just a matter of one became more dominant than the other depending on the condition of, supposedly the condition of the colors of the trees and the building. And she looked at me and she said, well, then that doesn't explain anything. Now, in fairness, I've taught enough students to know that sometimes what comes out of their mouth is not what came out of my mouth. So I'm not going to say that's absolutely the way that the instructor taught it, but it was certainly the way she understood it, because if it was a situation where both the black and white moth were there to begin with, then what's the point? All you've shown is that you can have a population change of more dominant black or more dominant white, but it doesn't explain anything. And certainly we would say, yeah, you would expect that if there's some kind of environmental condition where the white moth cannot camouflage or protect itself very well, it's going to be more heavily preyed upon. No big surprise there. So what? Yeah, big revelation there. Okay? And yet this is still considered one of the classic examples of evolutionary action involving, quote, natural selection. And one can certainly surmise if this is all you got, then you haven't got much. Okay, now, I mentioned, even by Dawkins' own definition of natural selection, not saying that I agree with that, but we're going ahead and using it because it still works for us as an illustration. Even by his own definition and explanation of natural selection, you still have to get the trait into the population. Okay? According to Lynch, in his book in 2007, he suggests there's only really 3 ways you get traits into a population: mutation, recombination, and random genetic drift. Okay? I'm not going to talk much about random genetic drift, okay, which actually, in a sense, involves recombination and mutation anyway. And I'm not going to talk a lot about recombination because, in some sense, it still doesn't explain where it originated, just explain how it moved around some. So mutation ultimately is where we come from. If we're going to develop a brand new trait that hasn't existed before, we've got to change something in the DNA, and that's going to involve mutation. So we're going to focus on mutations as example of getting new traits. See, and Jay's not the only one that can Photoshop. All right, so change in DNA sequence is how we're going to simply define mutation. Remember, yesterday I talked about the A and the T and the C and the G. That's the letters that make up the DNA sequence. We change a T to a C, that's a mutation. We change an A to a G, that's a mutation. Okay, so that's what we're going to simply define as mutations. Anything that changes those letters, that's a mutation. Thereby, potentially, when you change an A to a T, you have potentially made some change to the DNA information. Now, what that change is, sometimes very hard to tell, but you potentially made some change. And in that potential change, you have now the potential of a new trait that natural selection can now work upon. Okay, so let's talk specifically about what would be termed beneficial mutations. For a number of years, some creationists were arguing there's no such thing as beneficial mutations. But see, again, it's a matter of syntax. Okay, if we define a beneficial mutation as simply a mutation that makes an organism better able to survive in a given environment or a given condition, then there are clearly beneficial mutations. So if we have a mutation, WT stands for wild type. That's our original sequence. Mutation, whatever it may be, happens to the organism. It will have some effect. It'll either make the organism more fit, less fit, or what we call neutral. Although, as we learned, neutral is actually kind of more like near neutral, and they build up and such, and especially in humans, you're degenerating. You're just a bunch of mutants running around. All right. Antibiotic resistance is the classic that evolutionists talk about. You have a population of bacteria, you throw in an antibiotic, and most of them die. But not always all of them die. Sometimes some of them survive. Now, there's various reasons for this, but we're going to look at the mutational reasons in a second. And this is one of the reasons why it's usually important to take all the antibiotics the doctor told you to take. Because if you stop short because you're just feeling better, then you may have more likely left some that should have been killed, and they're still there. Okay? But sometimes some survive. I spent 20 years working with antibiotic resistance mutations and such when I was back years ago, younger, actually putting in 24, sometimes 25 hours a day in the lab and such. And my wife made me wear a name tag so that the kids would recognize me and all that stuff. All right. In 2005, I wrote a paper that was published in the Creation Research Society Quarterly, which is our quarterly for the society. If you're not a member of the society, by the way, you need to be. Okay? Just put that plug in. All right? I wrote a paper in the Creation Research Society Quarterly talking about antibiotic resistance, trying, in my opinion, to lay it to rest once and for all that this is not giving the evolutionists what they're trying to get. Okay, you have the antibiotic present. The organism is resistant. How is it resistant? Well, if we look, what happens? Loss of enzymatic activity. SOS response is halted. Loss of regulatory protein. Loss of a poron. Reduced affinity. Loss, reduced, loss, reduced, loss, loss, reduced. See the trend here? What's the trend? The trend is that the organism is getting rid of or minimizing a pre-existing activity, be it a transport, be it an enzyme activity, be it replication, whatever the case may be. It's reducing or eliminating that trait. And in so doing, that now gives it the ability of resisting the effect of that particular antibiotic. And different antibiotics act differently, so the organism has to do something different to become resistant, such as kanamycin resistance. See, if I reduce the formation of a transport protein, the kanamycin cannot get into the cell to begin with. Therefore, it can't kill me. But what have I done? I have reduced the formation of that pre-existing transport protein. Okay? So does that explain where the protein came from? No, of course not. And yet this is a classic example of evolution, saying, see, this is evolution in action. What? By getting rid of systems? Right? More evolution like that, and we'd all just be puddles of jelly on the floor. Right? But this is exactly what antibiotic resistance gives you, is these kind of changes in the organism. They're clearly changes, and they're clearly beneficial to the organism. If you are a bacterium, if you can imagine for yourself as a bacterium, okay, and here comes an antibiotic, then you would kind of like to have the ability of doing this that makes you resistant. Right? It's a little bit like your foot's caught in the bear trap, and here comes the bear. What are you going to do? Yeah, you're going to chop off your foot. Now, here's the key thing. See, the bacteria can grow their foot back. You can't. So they're quite willing to make this sacrifice temporarily. And then when the antibiotic is gone, then they'll restore the system. But you cannot use that as an example of how it originated to begin with. Losing a transport protein or losing an enzyme is not an example of where the enzyme or transport protein came from to begin with. And yet, as I reiterate, this is classic example that evolutionists will teach of examples of evolution and natural selection in action. Okay? Let's look at human mutations, because people kind of like to talk about humans and how much of a mutant we are. Okay? Some 4 to 6 million years ago, or maybe 8, or maybe 10, or maybe 12, depending upon what they finally settle on, because it keeps changing all the time. But supposedly some 4 to 6 million years ago, we and chimpanzees had a common ancestor, and our evolutionary lineages diverted from that. And in these last 4 to 6 million years, we have developed a number of mutations that now make us human, as opposed to another type of primate. Okay? Now, clearly those would be what we call beneficial mutations, because they're benefiting us, making us human, not chimpanzee, gorilla, orangutan, whatever. All right, so let's take as a beneficial mutation HIV infection. Okay, HIV, the virus HIV, will infect what is called your CD4 plus T cell. That's an immune cell. It'll infect that cell by binding to a protein on that cell called CCR5. It binds that protein, infects the cell, kills the cell. And if it becomes prominent enough in your body, you get what would be called AIDS, acquired immunodeficiency. It affects your immune system. There is a small percentage of humans that lack that protein because of a deletion mutation. Deletion mutation of the DNA, that protein, that CCR5 protein, is no longer made. If it's no longer made, then there's not a binding site for the HIV virus to attach to. And they're resistant to HIV. Now, if you're exposed to HIV, then this would clearly be a beneficial mutation. But it's at the loss of a pre-existing protein. Does this mutation explain how the CCR5 protein evolved to begin with? No, clearly not. It's beneficial if you are exposed to HIV, but it certainly doesn't explain the evolutionary development of anything in the human, just the loss. Okay, let's take another example. Right? Malaria resistance. Sometimes classic examples offered are sickle cell anemia. But this is actually a better example. Okay? You have on your red blood cells what's called the Duffy antigen. And if you're Duffy negative, it gives you a greater resistance to malaria than Duffy positive. Okay, let me illustrate that. Here's your red blood cell. It has the Duffy peptide on the surface of it. That is the peptide that the malaria protozoan will bind to, infecting the red blood cell, giving you malaria. But in some people, because of mutation, the Duffy peptide, the Duffy protein, is no longer made. Therefore, the malaria has no site to bind to, so it does not infect the red blood cell. So if you're exposed to malaria, then the absence of the Duffy peptide is beneficial. And it's certainly better than having sickle cell anemia, because as far as we can tell, there's not any serious consequence of loss of the Duffy peptide. But once again, what's our illustration? It's beneficial, but at the loss of a pre-existing system, in this case, a protein on the surface of red blood cells. So do we use this as an example of how that protein evolved to begin with? It fits the classic picture Dawkins talked about, natural selection, acting upon traits that are already present in the organism, and you have benefit, and the organism is better able to survive. In this case, humans without the Duffy peptide survive better in areas where there's malaria than those that have the Duffy peptide. So there's going to be a selection for that. But what do we have going on? It is at the loss of a pre-existing protein. Okay, let's do another example. Human lactose tolerance. How many of you are not allergic to milk? Now, most. And the reason is because you probably have Northern European descendants. If your descendants were from almost anywhere else in the world, you would likely be allergic to milk. Okay? The wild type, the original, what Adam and Eve were. When you're a child, you make the enzyme lactase, which breaks down the lactose, which is the milk carbohydrate. But as you move from child into post-puberty, there is a blocking action from a repressor protein that comes from the MCM6 gene, and it blocks the lactase enzyme, and you don't make lactase. Therefore, as an adult, you cannot digest the sugar in milk. So you'd be what we call lactose intolerant. But for most of us here, and I'm glad I am one of them because I enjoy milk, our descendants lived in Northern Europe, most of them, and they, as part of their culture, raised and milked cattle. And so milk became part of their diet. A mutation was acquired in the MCM6 gene that kept it from blocking the lactase enzyme gene, and you still make lactase. But it is a mutation. So you're mutants. Now, it's beneficial if you like milk, but it's beneficial also if milk is very available as a food source. But once again, what's happening? It is because of the lack of a pre-existing regulatory system. So what we've done is we've knocked something out that was there previously. So do we offer this as an example of how we evolved lactase utilization to begin with? No. It's an example of losing a pre-existing regulatory feature. So we'll use the analogy of a house. Let's say in your house, you have your dining room, and a separating wall then, on the other side of the wall is your rec room. But your wife, being the social magnet and social queen of Milwaukee that she is, she wants to have a bigger dining room so she can throw bigger parties. And you look at the structure of your house and you realize the only way you can achieve that without totally rebuilding the house, and you look at your bank account and realize that's not going to happen, okay, is you knock out that dividing wall. Okay? You lose your rec room, but your wife now has a bigger dining room, and she's happy, and everybody knows if she's happy, everybody's happy. Okay? But what was it achieved by? The loss of a pre-existing wall structure. Would you tell a carpenter that the way that you build a house is by knocking out a wall? They'd look at you like you are obviously some kind of spatial stupid. But that's exactly what we're being told. That's exactly what students are being taught in biology classes, is these examples of knocking out an existing wall and saying, see, it's an evolution, wonderful how it is able to achieve all these great constructs, and it's not constructing anything. What we really have a situation where, in terms of just generic biological complexity, evolution claims that everything is moving upward, climbing Mount Improbable. But see, creationists say, no, we start at the top of Mount Improbable and we're coming down. And the mutation examples illustrate that. Beneficial, yes, under certain conditions, but they're at the expense of pre-existing. You came down off the mountain. You didn't go up the mountain. Okay? Chromosome design. Go back to this for just a moment to look at the sophistication we're seeing in biological systems. Okay? Many promoters, we touched on this a little bit yesterday, many promoters, those are the start points for gene action. They require a combination of one or more chromosome areas or even one or more chromosomes coming together. So for the gene to actually function, we can't just, remember linear code? No, it's not linear code. You have to actually, in three-dimensional fashion, bring pieces of DNA together to get it to even function. Therefore, as we mentioned yesterday, if you start having mutational changes and deletions and such in these pieces of DNA that would make them jump, they'll no longer function to be able to form this kind of three-dimensional structure. But let's go even a step further. In the cells in your body, in your liver cells, in your kidney cells, in your heart cells, in your brain cells, you have the same DNA. But yet, liver cell doesn't do the same thing the heart cell does. Heart cell doesn't do the same thing the brain cell does. They got the same DNA. Why don't they do the same thing? Because they don't use the DNA in the same manner. Even though it's the same DNA, they don't use it in the same manner. And one of the ways is because, remember I talked about three-dimensional structure acting in a four-dimensional dynamic? That four-dimensional dynamic makes the liver cell work and read the DNA differently than it makes the brain cell read it. So we have this constantly varying among different kinds of cells in your body with the same DNA. And do we understand all this yet? No, we don't understand it all yet. How the liver cell fully knows it's a liver cell versus a brain cell. We don't fully understand how that is achieved yet. But we do know the liver cell says, I'm going to take this DNA and bend it this way. And the brain cell says, I'm going to take that same piece of DNA and bend it this way. We don't fully understand how they know they're supposed to do that, but that's what they do, and that's what makes them different. It's called differentiation. Okay? Also, in your chromosomes, you have acoustic vibration. I think if you listen, you can probably hear some music going on. The chromosome strands actually vibrate 10 to 100 cycles a second. This gives us what we call an open structure or a euchromatin structure. By keeping that vibration going, it allows proteins to slip in that wouldn't be able to slip in otherwise. Figure out how to design that. Okay? The central dogma of genetics. This goes back to Crick and Watson. Central dogma, DNA gene. Remember, we don't any longer really know how to define a gene. But we'll go back to the archaic term gene. DNA gene undergoes a process called transcription, and a copy is made from that gene that is actually RNA. RNA has a structural difference than DNA. And then that RNA is used in a process called translation to provide the information to make a protein. That's the central dogma. Now, let's complicate it a little bit, because I always tell my biology students, you want things to be too simple. What would be the challenge in that? And I think sometimes God didn't want it to be too simple, because then we might actually think that Darwin was right. The truth of the matter is, I think in some ways, God put in such marvelous design that we will, as the book of Romans says, literally stand before him a special kind of stupid, that we didn't recognize his finger. Okay. Now we understand what you make from the DNA is actually not yet messenger RNA. It's what we call pre-messenger RNA. And you cut segments out called introns, and what you have left is called an exon. Now, the introns are not necessarily wasted. They have a whole other function we don't have time to go into. But the exons, then, will arrange themselves in such a way, splicing themselves together, or being spliced together, that now you have the messenger RNA, and now the process translation gives you a protein. But let's take the very same segment, the very same exons. We don't have to do the same thing. We can rearrange them. So now they're in a different arrangement. And now we get a different protein from the same DNA, from the same RNA, but now a different protein. Okay? But let's not stop there. Let's again take our exons. We'll have an exon that will actually, instead of being translated by the ribosome, it will actually interact with the proteins in the ribosome, binding to it, causing the ribosome to slow or stall when necessary as part of a way of regulating ribosome activity during translation. So the messenger RNA exons are not always just for translation. They themselves regulate. But let's not stop there. With many messenger RNAs, it's not just the linear code. You took biology and learned that linear code where you got codons. AAA means a certain amino acid, and UUU means a certain amino acid. That's the linear code. But it's not just the linear code, because the messenger RNA also folds into these three-dimensional structures. Now we're talking about RNA, not even DNA. The RNA folds into a three-dimensional structure, and we know this is important because two different folding structures of messenger RNA will give us two entirely different proteins. Just based on how they fold, the sequence can be entirely the same. But based on how it folds, we get a different protein. And in fact, as I state here, we now are understanding the disruption of these folds can actually cause certain diseases. So we now recognize that some diseases in humans are not because of any change to the DNA sequence, but because of changes in how the RNA is folding. And here we have two examples of how the same sequences of RNA can fold. Okay? Most known genetic diseases have been found to be associated with what's called the non-coding regions. In other words, that I mentioned yesterday, the junk. We're now finding that most genetic diseases are actually associated with the junk. Well, if it's junk, then that shouldn't be the case. Because remember, it shouldn't make any difference what happens with the junk because you're not using it anyway. So why would it be the genetic diseases are associated with the junk if the junk is junk? Follow the junk? Okay. But even within the linear code itself, there's still a second code. There's what are called duans, where a particular codon, like say ACA, may serve both for translation by the ribosome making a protein, but also serve as a site for transcription factors to bind to. So when you would change a mutation in that codon, which is what evolution would suggest will happen as you're building new proteins, so you can evolve upward, going from fish to amphibian, etc., grow wings and fly. Right? But you also now are affecting these factors' ability to bind. And so maybe you say, okay, if I make a mutation where this codon changes sequence, so I now can grow wings, but the transcription factor says, yeah, but I don't like that change. So now you've interfered with me being able to do what I need to do. And so now you're running in a situation where you're telling the captain, the ship is sinking, but the engines have never run so good. Because you have to have it all working together. And you can't benefit on one hand and not benefit on another, and the system still work. And it's becoming more and more clear that that's the case. Okay? Here we have two sequences exactly the same. Different information? Well, maybe, if they fold differently, but it's even simpler than that. All I have to do is put what organic chemists would call a methyl group on one of the guanines. And voila, I've now changed the information content of that strand of DNA. I didn't change the sequence at all. I just did what's called methylation. Now this fits into a whole area called epigenetics that I simply don't have time to get into. But we see that we can do modifications to the DNA very simple. They don't even involve sequence changes, and yet we still dramatically change the information content of the DNA. Okay, this is one of my favorites. Okay, we'll start out as an example. This is repairing bacteria. We've been talking about humans. We're going to go to bacteria for just a second to show this example. Okay, mut-Y is a repair protein. All organisms, bacteria, human, everything in between, you have proteins, you have enzymes that are responsible for repairing errors and damage in your DNA. And in the bacteria, mut-Y is one of those proteins. It finds errors in the DNA and repairs them. In fact, you're dependent very heavily upon that in your body because you're constantly undergoing DNA damage in your cells. And so you're very dependent upon the ability of these repair systems to catch that and repair it. Otherwise, you'd probably died a long time ago. Okay, there's another protein called Endo-3, and it releases an enzyme that traverses its way all the way down a long strand of DNA. And if it hits the mut-Y, then the mut-Y releases. Now, what becomes significant about this is in bacteria, there's not very many mut-Ys. And I thought, wow, how can these so few mut-Ys scan and do surveillance of the entire chromosome? Well, this is how. Because by getting hit by that electron, that told the mut-Y there's no mutations in that strand of the DNA, so it knocks off and goes to a different place on the chromosome and starts again. So in essence, the electron did the scanning for the mut-Y. The electron moves much faster. You're able to do that very quickly. You want to see that again? I just love this. See? If it makes it all the way down, it hits the mut Y. That's a signal mut Y. There's no mutations in this region of the DNA. Drop off, go to another region, and do surveillance there. Now, let's say that we do have a mutation, a mismatch in this case. We don't have the right base pair. Okay, what will happen now is when the Endo3 releases the electron, it'll hit that mismatch, and that'll absorb it. Now the mut Y is not hit by an electron, so it just keeps going down the DNA until it encounters the mismatch and repairs it. In humans, you don't have mut Y, you have what's called mut YH. We don't know if this is what happens in humans, but your mut YH has many chemical characteristics very similar to bacterial mut Y. So it may well also be done in humans as well. I'm voting that it probably is, because it's so fun. You know, how could God not do something that fun in humans? Book just published called Information Processing at the Cellular Level Beyond the Central Dogma. Remember that central dogma? DNA is transcribed to RNA, which is translated to protein. Okay? Albert Riva says that while the central dogma has always been an oversimplified model, we've always known that. It's always been an oversimplified model that did not account for a multitude of other processes occurring inside the cell. Its limitations are today more dramatically apparent than ever. Remember, we're moving farther from chance, farther from Darwinian explanations of what we see. It has become clear, he says, that the overall picture is much more complex than the one outlined by the dogma. Have you ever noticed something? Is anything ever less complex than we previously thought? See, that's why it's moving farther and farther beyond the reach of chance. Because, like I said, Darwinian type thinking, evolution, really even any evolutionary type thinking, was fertilized and propagated at a time in science when they did not understand many of the basic principles, let alone the sophisticated aspects of biology. And that kind of vacuum, if you will, that kind of void in knowledge allowed all kinds of speculations to come in that sounded true, that sounded okay, because among other things, let's face it, it gave us the opportunity, as Lyell said, to get rid of Moses, to get rid of the writings of the Bible. Okay? So what we're faced with from a evolutionary perspective is when we look at the symphony of life, which is a good way to, and this is the way I describe it to my biology students, the symphony of life. Look at the symphony of life. We are stuck with an orchestra with no conductor and a symphony with no composer. ORFan genes. They appear suddenly in the biological world. And while ORF stands for Open Reading Frame as part of how we define a gene, it also is a play on words because it is an orphan. We don't know where this gene came from. It doesn't appear to have any ancestry. Okay? And this directly contradicts Darwinian common descent, because all genes should have ancestry by Darwinian common descent. Okay? So what we would say is the Darwinian prediction would be you have gene, give all the capital A's, and then as it's evolving, see, we can follow that there's slowly some alterations to it. But we can always go back and figure out where it came from to begin with. And this is classic of what Darwinian evolution would expect in evolution of a gene. And we see its Darwinian lineage, then its evolutionary lineage. But an orphan gene, it didn't come from anywhere. It's just there. There's no evolutionary lineage to it. Quoting from a 2003 paper, if evolution works through descent with modification, why is it that we do not find today any of the necessary intermediate sequences that must have given rise to these orphan genes? If Darwinism is true, then the one on the left should be true. But instead, we find many, many genes that are orphan genes. We can't explain that from Darwinian explanations. Okay? In fact, orphan genes are found in every taxonomic group studied. It's not like they're just in one or two types of organisms. They're in every taxonomic group studied. And they comprise some 10 to 30% of the entire genome. So it's not just one or two. 10 to 30% are orphans. orphan genes are also just as likely to be essential as the, quote, old genes, the genes that they do say they can follow an evolutionary lineage to. So it's not like that suddenly this new gene comes into play, but it yet is trying to establish its importance. It's just as likely to be important as genes that would have some kind of evolutionary lineage to other organisms. So it's not a matter of new genes coming on, but they're not yet essential to the organism because the organism hasn't necessarily evolved to that point yet. New gene in this organism, and it's just as essential as any other gene in the organism. Okay, so what's the origin of orphan genes? This is clearly a big question for the evolutionist. Right, well, what they're going to say is that you have, what's that? Junk DNA is undergoing some unknown adaptive mutations and becoming this gene. That's why you didn't see it there in an evolutionary lineage, because it evolved from not a previously existing gene, it evolved from junk. But what's the problem? Among other things, what's the problem of junk DNA? You don't have enough junk DNA. They're losing the junk DNA. So not only are they losing the junk DNA as a repository for all the mutations going into the human chromosome, but now they're losing junk DNA as a source of DNA to make these orphan genes. So that's why junk DNA is so vital to them, because they've got to have this. If they lose junk DNA, then they have all kinds of problems that they simply won't ever want to admit. Okay? Also, remember what we've talked already, just in brief, about with mutations. Do mutations explain the origin of? No. You have to start out, and the mutation actually removes it, takes it away, even if it's, quote, beneficial. So they just pretend that adaptive mutations would actually account for an orphan gene. But what we see illustration-wise over and over again is that mutations don't give you that. So we have two problems that they're trying to, we have two problems with the scenario they're trying to propose. Okay, in the human, because people want, what is going on? They don't care so much about fruit flies and bacteria and guppies and such. They want to know what's going on in human. In human, found 1,177 what are called putative orphans in humans with no detected counterpart in other primates. In other words, no evolutionary lineage. That would seem to be the classic definition of an orphan gene. But the authors of this paper say if these orphans represent valid human protein coding genes, in other words, if they're valid genes, we would have to conclude that the vast majority were born after the divergence from chimpanzee. In other words, that 4 to 6 million years since we diverted from chimpanzee. Okay? Such a model would require a prodigious rate of gene birth and a ferocious rate of gene death, erasing the huge number of genes born before the divergence from chimpanzee. So in other words, if those 1,177, 76 orphans that they detected in humans were real, then in the 6 million years since our common ancestor, they would have eliminated most of the genes that actually make us human. And they realize that's not going to work, so here's what they do. We reject such a model as wholly implausible. Now, they don't have any evidence that they're not truly orphan genes. They just simply say, it does not fit evolution, therefore we reject it. So we have a clear example where the paradigm drives the conclusion. We reject such a model as wholly implausible. We thus conclude the vast majority of orphans are simply randomly occurring sequences. No data to support this. But they're forced to conclude it because their paradigm is evolution. Evolution will not account for it. Therefore, they have to conclude that they're not truly genes. They're just randomly occurring sequences. Even if they have every characteristic of a gene. Walks like a duck, talks like a duck, looks like a duck, but it's not a duck because my model will not allow for a duck. I think the better illustration is a black swan. No such thing as black swans. Here's a black swan. Well, I can't account for that. They don't really exist. See? That's the approach these authors took. Now, since then, there's only really been, that I've been able to find, one other study dealing with orphans in humans, and they found 60 orphans that they did very much prove are not randomly occurring sequences, but are functional genes. And I think as time goes on, the other 1,100 will also be found, and probably more, as true orphan genes. Therefore, bringing back the problem that caused them to reject it, and that is there had to be a ferocious rate of gene death and a prodigious rate of gene birth, which clearly does not fit evolutionary examples, evolutionary data, but it's what they'd be faced with. So, in this, I referenced this, or I commented about this quote yesterday. I'll give it to you today. The phylogeny of the human genome resembles a patchwork of individual regions following their own genealogy. Does that sound like Darwinism? No. A patchwork of individual regions following their own genealogy. In other words, we cannot trace your evolutionary lineage in your genome because it's too hodgepodge. Now, evolutionists have a number of explanations for this, but none of the explanations really make much sense. It's just desperate attempts to try to somehow piece the picture still together. Okay. A couple other little points. Study that came out just last year, 73% of all mutations in protein-coding genes. So 73% of all mutations in your genes and 86% of all predicted negative mutations occurred in the last 5 billion years? No, 5,000 to 10,000 years. In other words, the mutations that are in essence making you and not a primate occurred in the last 5,000 years. And this is not in a creation journal. This is in the journal Nature, which is about as anti-creation as you can get. Another study done in 2004. The genealogical ancestor of all present-day people. Now, they're not saying that there wasn't anyone else alive. They're just saying that we descended from this genealogical ancestor. Right? Lived when? 3 million years ago? 3 billion years ago? 3000 BC. Again, in the journal Nature. Not a creation journal by any means, but in studying the genetics of humans, they had to recognize the human genome is young. I talk to geologists all the time about how old the Earth is and how old life is. Well, that may be, but humans are very young. How do we know? Because we know genetically, which is a whole lot better, in my opinion, than your data, because we can actually look at this in the laboratory. So I think we're left with this. That's not how it works. It's not how any of this works. Now, lest you think, well, this is just some knothead, one of those lunatic, spatial kind of stupid creationists standing up there saying that, I point you to a website called The Third Way. The Third Way is a website initiated by people such as James Shapiro, a very brilliant microbiologist, but he's an atheist. He's rejected neo-Darwinian explanations for years, but he's not going to suddenly become a creationist. The reason I point to this is because I think this is very real. It's important to us, but it's also important to us to recognize other aspects of this as well. What they're challenging is that they reject creation because, of course, that's not their worldview. They reject neo-Darwinism because it simply doesn't fit the data. So they say we have to find a third way. And I would challenge that this is going to gain momentum. In fact, I've gone on record as saying that I predict 30 years from now what is taught in the biology classrooms is not going to be the Darwinism that's taught today. Why? Because James Shapiro would agree with 95% of what I just said today. But he's not a creationist. But he will admit that's true, that's true, that's true. It's not leading him to become a creationist, though. It's leading him to search for a third way. So while we creationists would love to think that if we can get people convinced this marvelous design that's unexplainable by Darwinism has to lead you to the creating hand of God, see, it's not about this, it's about this. So if I'm not willing to accept God, I'm going to what? Look for that third way. And this is, like I say, going to gain momentum. This is the direction it's going to go. I don't know what the third way is going to be. I'm sure it's going to be something that we'll still scratch our head at and say, well, that's no more reasonable than Darwinism. Because anytime man proposes something to replace God, it's not very reasonable. But that doesn't mean that they won't do it. And with that, once again, I'll put a shameless plug in for my video. And I'll close. We have, I guess, maybe a few minutes for questions. All the questions yesterday. Yes?
With the mutations that are happening that we can observe now, as a layman to all this, it seems like they should be causing
Well, many of them are.
Oh, they are.
Yeah, remember I mentioned that, for example, there's many mutations now in what was called the junk DNA that are now linking to genetic diseases. For exactly because of that. Because when they cause the DNA to fold differently or they interfere with some kind of protein binding, that leads to a genetic disease. So they are, in fact. Now, one of the problems of trying to always identify, and Jerry could probably talk more elaborate on this than I can, but one of the problems of always trying to identify the cause of genetic diseases is it's not always just a single identifiable mutation. And it's not always even recognizable if it is a single identifiable mutation because we're still putting together what is the true, quote, wild type sequence of humans. Because, see, everybody's DNA differs a little bit, but is that T in the location on your DNA and the C in the location of my DNA something that's actually going to cause a disease in me at some point in time or not? We don't know. Okay, so there's just, it's still, the complication of it is still such that it's very much a struggle to always identify what is causing a particular genetic disease. Yeah?
Ever since the time of Darwin, the missing links in the fossil record has been recognized as a major problem. Do you believe that the missing links in the genealogical record or in the genetic record might be even more of a problem?
I think it will. Two things in answer to that. First, more people are familiar and can understand the fossil data than are really familiar and can understand the genetic data. And as Jay was talking a little bit about this morning, a lot of scientists, I think you were talking about this morning, anyway, maybe I'll give you tribute to something you didn't say, but a lot of scientists, they understand the weaknesses in their particular field. But they assume everyone else has got it all figured out. And therefore, just because I can't explain it in my field doesn't mean that suddenly Darwinism isn't true. As I've stated several times, most scientists are evolutionists not by decision but by default. That's all they know. That's all they've been taught. They've never considered looking at something else. And in fact, the very effective propaganda that evolutionists have done against creation keeps most of them from even being willing to give it any kind of hearing whatsoever. Oh, it's just creation, just immediately won't even listen to it. And when such happens, then they don't expose themselves to the arguments, and therefore that's all they know is evolution. Must be true. Whether I understand it all, whether it even makes sense to me or not, it must be true. Right? In fact, I was going to bring another quote, but I don't have it on this. If you come to my breakout session tomorrow, I have a beautiful quote that by an evolutionist says exactly that. It doesn't make any difference how crazy it is. We have to accept it because the alternative is creation, and that's just not acceptable. Yeah, that's the quote. That's it. Yep. Next question. Any other questions? Still digesting? Yes.
I missed one of your later slides. You used the word phylogeny, and I thought
phylogeny really just is referring to, in broad categories, really referring to your evolutionary history. See, the human phylogeny would be our relationship with primates and with other animals, with other mammals, and the relationship of mammals with, birds and reptiles and such. And so that's what we mean by your phylogeny. It's really just a classification terminology. Other questions? Yes?
You mentioned that a lot of the mutations are very low-key. Has there been any kind of a study or a look at exactly how significant the mutation has to be before it gives a reproductive edge? When you look at the things that happen, that we see as God's hand, some chance.
The difficulty in that question is it depends on the system and it depends on the environmental conditions. Let's use Linsky's bacterial work as a classic example, because that's what evolution, they hold it up as a classic example. If you're not familiar with Richard Linsky, he took the bacteria Escherichia coli and grew it for over 50,000 generations in the same never-changing condition. Never-changing condition to look exactly that. What kind of mutations does it take for this organism now to become more fit, more adapt? His only criteria was if it grew faster. which is typically in bacteria what you'd look for. Did it grow faster? And he found several mutants that grew faster than the original. Okay, what type of mutations did it take? Well, in the examples that he found, it was a matter of truncation. They would eliminate parts of the chromosome or shut down parts of the chromosome so they could simplify their metabolism, simplify their reproduction, which would make sense. That would allow them to grow faster because they don't need, if they've been in this particular never-changing environment for 25,000 generations and I've never used my flagella proteins once, why do I need them for? So if I get rid of them, by mutation, those are deleted out. It actually is beneficial to me in that now I have less chromosome to maintain so I can actually replicate faster. It takes 40 minutes to replicate the wild-type E. coli chromosome. If I eliminate some components, I can maybe do it in 39.5 minutes instead of 40. That's a 30-second advantage spread out over 1,000 generations. That's a major advantage. And those are kind of mutations he found, up to the last mutation, which is this famous citrate mutation. And all that really was just folding together of two promoter sites. So it wasn't near as complicated or sophisticated as he was claiming it was. So the answer to your question is it depends on the organism, it depends on the condition. Sometimes it doesn't take very much at all to make a significant impact. And sometimes even what you'd think this is a major mutation and the organism doesn't seem to be affected by it at all. Yes?
There's a statement by evolutionists that goes like this. Progeny recapitulates phenology. Is that what you were getting at?
Not really. I mean, in a loose sense, it could be. What they're referring to, of course, is they're referring to embryological development, where the embryo, as human embryo, you went through your fish stage, your reptile stage, et cetera, which is, of course, all nonsense. But in a loose phase, it could be in that genetically we should technically be able to identify this is when you were a fish, this is when you were a reptile, this is when you were, early mammal and such. But a lot of that, you see, is by virtue of the junk DNA claim. Because you're no longer using your fish gill genes, therefore they've randomized by the time you're a human. And so we don't see them as fish gill genes anymore, we just see them as junk. And that's so, in essence, the junk DNA is the DNA version of the vestigial organ. You use this organ when you're a fish, you don't need it anymore, so it's just become a useless organ. Okay? You use this DNA when you're a fish, you don't use it anymore, so it's just become useless DNA. There we go.
Is it really? When it appears
Well, were you at my talk yesterday?
Yeah.
yeah.
No, I mean the junk. I mean, when it appears that the human embryo goes through these different...
Oh, the recapitulation.
Yeah.
No, what happened, right, what happened was...
My professor telling me this. That was a long time ago.
And it was nonsense then, and it's nonsense now. It was even at Haeckel, German embryologist, proposed that back, 1880, 1881, okay. Even at the time he proposed it, it was known that was not very accurate. But at that time, they didn't have some of the microscopic capability we have today to really go and look in detail. But even then, they realized he actually had faked some of the drawings. Okay, there's a nice paper, 1999 paper by Richardson, that went back and looked in detail at this. And really very, very accusative of this is utter nonsense. How could anybody have continued to promote this? And yet it's still in the textbooks. But he showed a picture of here's what the human embryo looks like, in the first 7 days, 14 days, 21 days, whatever. Here's what the fish embryo looks like. Nothing even close to the same. And that's been well documented. And yet, even today, you'll open up textbooks, and while they may not use that terminology, they're still trying to imply the same thing, that you will actually retrace your evolutionary history in your embryo. In fact, back when I was an undergraduate, a couple years ago, I was in an evolution class, and the teacher taught recapitulation. And at that time, I wasn't the student that was going to raise my hand and challenge everything right in the middle of the class, but I did go up afterward. And I said, okay, I'm a little confused because I really thought that embryology rejected the idea of recapitulation now. And he says, well, that's true, but it makes a good study piece for evolution. Yeah, exactly. And I was like, okay, I And those, sometimes you get a comment so left field you don't even know what to say. You're just kind of just dumbfounded. And then you walk away. And I thought, I don't think any of the students who was in the class this morning took it as a nice study piece, even though it's not true. As far as they were all concerned, it must be true. So, I mean, he was a nice guy, but literally it was a blatant lie.
How is it a good example?
Yeah, that's a good question. Yeah, exactly. Are you so desperate for examples that you got to make it up?
How do our young scientists going into the field even get through all this minutia? If they're given all this erroneous material, and then they have to start from...
They need to join the Creation Research Society. They need to get our publications. They need to communicate with us and help us because we plowed the road in front of them.
That must be the biggest disappointment of all is when they get into this material and find out that none of this is true.
But unfortunately, too many of them get impressed by it. They don't have the resources. They don't have the understanding that exactly that. And they assume, wow, it must be true. Must be the case. And therefore, they get pulled away. Right now, one of the things I'm doing is I'm reading books written by atheists. Not evolutionists, atheists that used to be Christians. And every time I say, what's their story? What's their story? And usually their story is, number one, they had some emotional trauma that they felt betrayed by the church. Right? Remember that. They had some emotional trauma they felt betrayed by the church. Church is judgmental. Okay? And number two, they began to be attracted to, well, this is what science says. Must be true. Must be true. They didn't have the resources or make available to themselves the resources to understand that wasn't true. Or there's a better explanation. And so, in talking with young people, you've got to not tread the territory alone. We're here to help. Society of Creation, Concordia College is here to help you traverse through this. I was talking to a young man just a week ago. He wants to be a geneticist. I said, I'm sorry, there's no easy way to do it. You're going to have to plow right into the university, right into Satan. I call it Satan's meat grinder. He's right there saying, come on, little kiddies. And you think about it. Your average 18-year-old, their brains are full of snot. Not even mush, it's snot. They don't have anything up there. And Satan's saying, come on, little kiddies, I'm so ready for you. When I'm done with you, you're just going to be all chewed up. And that's what he does. But you can traverse it. You can traverse it if you're prepared and you take advantage of the resources available. And we've plowed the road ahead of it. We know what's there. We know how to help you through. And there are answers to these questions. And so I was telling him, you will make it through. You will be tested by the fire. But when you come out, what is it the fire does to metal? It makes it sharper and stronger. Then you're ready to go. You go toe-to-toe with the evolutionists. They hate that. They hate that because you know what they know. And they don't like that at all. Yes?
Another answer to her question is that academic positions have such a draw. You have freedom. And if you even begin to question that, you're weeded out from the very beginning.
Yep.
Look, who's this? Bergman's.
Jerry's book, right there? Yeah.
What's the name of that book?
Part of the dissidents.
Yeah.
That's the answer. You don't have a voice anymore. You're rooted out of
Can't think of his name at the second. Anyway, he said in academia, well, you have your colleagues, running around sniffing each other's underwear, looking for any dissent from the party line. And literally that's what you have in academics, especially in this country. As one Chinese scientist said, in China, I can criticize Darwin all I want, but I can't criticize the government. Here you can criticize the government, but you don't dare criticize Darwin. And he said, you people aren't as free as you think you are. Yes?
I understand that genes can be linked to physical disorders and...
Sure.
But are they linked psychologically?
I might turn that question over to Jerry.
Yeah, they are.
They have...
Depression's a good study. Amish tend to have a problem with depression in the families, and that was one of the first studies they were able to determine because they intermarry. They don't rarely marry on the outside. So they found in studying them that one would commit suicide, and they found out the guy's grandfather committed suicide, and the guy's uncle committed suicide, all in one family, or largely in one family. So depression was one of the first things they looked at relative to genetics. And now they found many, many other conditions that likewise are genetically influenced and caused by influence. It doesn't cause you to go insane, but it predisposes you to insanity. Bipolar is another one that's clearly a genetic factor. So the idea is if you realize you have this genetic factor in your family, then you can work on overcoming it. You can realize it's there and deal with it so it doesn't take you over. A lot of people say genes determine things like that. They don't determine things like that, they influence things like that. Same issue you have with homosexuality and other sexual orientation. There are genes that influence it but don't cause it. It's like you don't inherit a gene to be a good basketball player, but inheriting a gene that makes you tall sure helps.
We're into your break time. Don't want you to miss your break. I'm going to put another shameless plug in for my own breakout session tomorrow. We're going to go in and follow this even further. It's called Rethinking Paleontology. Of all things, a geneticist challenging a paleontologist. That's what we're going to do. We're going to challenge the total paleontological paradigm because they're not geneticists and they don't understand things that can explain what they're seeing in a non-evolutionary manner. So that's what we're going to do tomorrow in my breakout station. So I guess we need to break, but I'll be around for questions. You had a question over here, so...
By 5 screening with your questions of the panel for tomorrow.
Right. Yeah.